So this webinar is really, we've talked a lot about what we can do with phylogenetics, and we've talked about some of our findings. This is not about that. This is an unapologetic introductory description of the phylogenies. So we found through the discussion that it's helpful to all get ourselves onto the same page. So that means those of you familiar with this material, just sit back and enjoy it. So the basic aim of this webinar is just to describe, really from first principles, what are these objects. So we've shown, at different PANGEA meetings, we've shown phylogenies such as this one that is generated with our own software, showing different viruses from within host and between host diversity. We've shown some conceptual phylogenies such as this one that we use to infer transmission, and we've shown some broad scale ones such as this one that is used to infer the migration of the virus across different regions of Africa and beyond. So what I want to do is just to link these phylogenies to a basic understanding of the replication process of HIV. And the first question is before discussing what a phylogeny is even to ask, why is it that we choose this way of relating or representing the relatedness between viruses? Why do we choose trees to represent the distance between viruses? When other forms of relatedness, so for example, if you think about social relatedness, we talk about social networks. When we look at similarity between individuals, we create clusters. And when we look at, for example, the distance between things, so for example, the distance between geographical location, we use maps. So why is it that we tend not to use networks, clusters, and maps for viral genetics, but we instead use trees? And the answer is simply because HIV is a biological replicator. So you're all familiar with the replication cycle of HIV, that it comes into the cell as a double-stranded RNA virus. It copies itself into the cell DNA, and then the cell starts producing new viral particles, typically hundreds or thousands of them, and in the process, the cell dies, having produced many more viral copies. So let's represent this in a very simple conceptual way, and we start the whole thing off with one single virus on day 0. And let's condense that entire replication cycle to just one virus making many copies of itself. So by that entire replication cycle, we know from sort of beautiful analyses of David Ho and George Shaw's group and so on in the 1990s, each replication cycle takes about 2 days. And through a replication cycle, the virus acquires approximately 1 mutation or more within its genome. So every time it replicates, it acquires a mutation on average. So what we do next is we think about tracing an ancestral lineage. So we're going to follow this process, but we don't keep track of all of the viruses. We only keep track of the viruses whose descendants survive to become part of the sample that we're studying. So most of the viruses that have come out of this process, I highlight in gray here, they're viruses that never make it into the sample. They either just don't get to infecting a cell, or they become very successful but not in the sample of interest. And the property of the lineage is that it's what evolves into our sample. So the green one is differentiated from the other ones by the fact that its descendants are part of the sample. And then by day 4, we'll get another round of replication, so we'll have more viruses. And we keep on going in this way. So again, from all of those viruses produced on day 4, there will be many descendants that are not part of the sample. But the green virus at day 4 has its descendants part of the sample, and this goes on, and you have a day 6. So this is a straightforward lineage where you'll keep on following the virus whose progeny will eventually become the sample. Now, what happens if, for example, you have a growing viral population or you have a broad enough sample? At some point, you will end up with more than one virus leaving descendants. So if we go back to our day 2 population, and we now look at what happens in our day 4 population, in this case, we actually have 2 of the viruses, which I'm highlighting here, leaving descendants in our sample. And these descendants, some of them will be descendants of one of the virus, and some of them will be descendants of the other virus. And then we keep on going forwards, as before, keeping track of the viruses that leave behind descendants. So throughout this entire process, what we're doing, the key concept is that tracing an ancestral lineage consists of keeping track of which viruses leave behind descendants in the sample. And therefore, from this particular perspective, we don't care about the viruses that don't leave behind descendants. So we ignore them. So we take all of these viruses highlighted in gray and we remove them. And so what we end up is this history of the viruses which have left behind descendants with some events. There's a time scale here, day 0, day 2, day 4, day 6, and so on. What we see here is that this has a split, and the split takes place on day 2, and that's where the single ancestral lineage splits into 2 ancestral lineages. If I'd started my investigation on day 6, and I was working my way back to the ancestor, then this event here, I wouldn't call it a split, I would call it a coalescence, because 2 different lineages coalesce down into a single lineage, and that coalescence would take place on day 2. So that's a perspective you get from looking backwards at evolution. Now, what makes this complicated is that we don't observe this whole process. We usually infer this process indirectly from a sample. We very rarely get the opportunity to watch evolution except in experimental settings. So usually we only have 2 viruses in the sample, and I'm just going to talk about a very conceptual study, a study which consists of only 2 viruses, and from these 2 viruses we infer this history. Now, this object, where I've only kept track of the ancestral lineages, is in fact the phylogeny of the 2 sampled viruses. And the task of phylogenetics is to infer this phylogeny from data that you obtain only from these 2 samples in the boxes here. So here's some terminology for this very simple phylogeny. Is we have the sample, which are also known as the tips of the tree. So we think of this as a tree lying sideways, and like a tree, it has a root, and you branch outwards from the root all the way until you get the tips or the leaves, sometimes called the leaves. And our sample is always at the tips of the tree, and right at the bottom, everything always collapses down to a single ancestor, and that's the most recent common ancestor of the whole sample, also known as a root. And however complicated the phylogeny, this root is a single viral particle that had existed at some point in the past. The splits are the sort of branching event, and they have in this case, they occur on day 2. So the moment when the lineage splits is also known as the internal node, and because we infer these things from backwards from the present time, it's also sometimes known as a coalescence of 2 present-day lineages. So now if we look back at the first of the phylogenies that I said we would sort of go through and understand conceptually, we can now understand what we're saying about the viruses from person A and from person B. We have 9 viruses that we've taken in a present-day sample, and we work out what all of the ancestral viruses were at an earlier time. So each point on these lines is an actual virus that existed at some point in the past. And when 2 lineages coalesce, that means that these viruses actually have a common ancestor from this point. and at some point one of the descendants became this virus, or kept on replicating until its descendant became this virus, and the other ancestor kept on going until its descendant became this virus. Now, this is simple enough, but as trees get more complicated, correctly interpreting trees is not something which is difficult, but it is something which, if you're not familiar with it, requires a bit of practice. And the reason it requires practice is not because it's difficult, it's because they're visually misleading. So if we look at these viruses, and I've highlighted 3 of the 9 viruses in this sample, and I've labeled them virus 1, virus 2, virus 3, and you can ask yourself which are the most closely related of these viruses. I'll just pause for a moment. I'll go through the answer. I'll just pause for a moment while I consider that question. Which are the most closely related of these viruses?
Directionality. Everything. Yeah.
So the answer is that the most closely related of these viruses are virus 2 and virus 3, because if I go backwards in time to find the common ancestor, there's a node here, which is the common ancestor of virus 2, and which is also, following this route here, the common ancestor of virus 3. So virus 2 and 3 are more closely related to each other than either of them is to virus 1. And therefore you expect virus 2 and virus 3 to be more genetically similar to each other than either of them is to virus 1. So what makes the phylogeny uncertain is that we don't observe it. Instead, we infer it from data collected only at the tips. So I mentioned this before. So how do we do this? Well, the way we do it is that as the virus goes through this replication process, and as you follow a branch, the virus isn't just replicating, but it's also mutating. So if I redraw the diagram, the biological diagram, throughout my 6 days, in this case I have the virus replicating, and every time, as I mentioned, the descendants acquire some additional mutations. So if I imagine the genome, this is a very short genome, of my ancestral virus, and it's got 9 base pairs here, and this is the genetic code for it, ACC, ATG, AAC. Then if I look at these viruses, which I follow my lineage, then they've acquired one mutation. So here the 5th position has changed from a T to a G, and here in the virus at the bottom, the 2nd position has changed from a C to an A. Then in the next generation along, I follow this virus with this genotype along, and now the first A turns into a T, and then in the second, in the last replication, the second C turns into an A. If I follow the bottom lineage, here the 4th A turns into a C, and the next one turns into an A. And the idea of phylogenetics is essentially to look at these you only observe these genotypes in leaves, and essentially you have to infer a minimal or a parsimonious set of events which can explain how these viruses evolved from a common ancestor. Another bit of terminology. So these things we've called mutations, but the mutations which are on the ancestral line are called substitutions. They're special mutations. They're mutations that have the property that they've survived all the way from the common ancestor all the way to the genotypes that you've sampled. Why do we need a special term for substitutions versus mutations? That's because they're typically much lower. So if you look at HIV and you follow HIV along a lineage, it typically acquires roughly 10 substitutions per year, whereas the naive expectation is that it should be at least 150. Because as I mentioned, there's roughly 1 mutation per replication cycle, and there's roughly 150 or more replication cycles per year. So why is the substitution rate only 10 substitutions per year and not 150 or more? That's because the gray viruses and the green viruses are not identical to each other. Typically, the mutations are harmful. So most mutations are more mutations make it less likely that a virus will survive along the ancestral lineage. And there's a second effect, which is quite specific to HIV, which is that the virus, as it goes through this process, there's a small probability that the virus will get transcribed into a resting cell and will turn itself into a non-replicating latent state, which will last for many years. And because if that affects the ancestral lineage, then essentially the mutational process just gets stopped for several years, and that results in a much lower substitution rate overall. Now, how do you go about inferring these phylogenies? Well, you start by entering these genomes into phylogenetic software, and the phylogenetic algorithms, which are encoded in phylogenetic software, usually most software give you a choice of lots of different algorithms. They use molecular models of how the virus accumulates substitutions. So this is taking into account what we know about the biology of each organism, which tells us, that A to C mutations might be more likely than T to G, or indeed C to A mutations might be more likely than, and so on. So you end up with sort of two matrices, about 32 parameters, and you can make simplifying assumptions. And you can also take into account that mutations might be more likely in some parts of the genome. You can see here there was no mutations in the AAC motif at the end, so maybe you might get a better model if you allow that to be invariable and so on. Typical software which are used include PhyML, RexML, IQTree, Mr. Bayes, FastTree as well. Those are very popular software. So now let's look at a real example of phylogeny. So here we've got a phylogeny which is the same as every other phylogeny I've shown before. Each line, sorry, the slides are jumping. Each line represents a particular virus at some point in the past. The tips, which are here, the dots, are the actual viruses from our sample. We've colored the viruses according to which patient they were sampled from. And we've put a number because we had lots of viruses from each patient. And we've collapsed down if the viruses were genetically identical, we've only counted them once. And the number tells you how many of that particular virus of that particular type we found. Then we reconstruct the ancestor, and the ancestral node is given the letter which identifies the patient. So we have patient F, patient E, patient D, and so on. So this is the ancestral history of all of the viruses from 6 patients. The additional piece of information is a scale bar, which is in units of substitution per site in the genome. So if you have a branch this long, it tells you that on average, each position in the genome is 5% likely to have had a substitution. So that's a very detailed phylogeny looking at individual patients. Phylogenies are useful because they can provide information at different scales. Here's a scale bar again, the same 5% as you saw before. So this is showing you that the phylogeny is at a wider scale. And this is the ancestry of individual viruses. And just as before, you have every point on this line is a particular virus at some point in the past. So we have HIV 1 group M. So these are most of the viruses. These are representatives of most of the viruses which are in circulation, shown in red. Their ancestor is a single virus which is labeled A. And you can see that the other viruses that then share a common ancestor before that are HIV viruses from chimpanzees. There is a separate type of HIV1 called group N, and that roots back, its ancestor is B here, and it's the next ancestor along, if you go backwards in the tree, takes you back to a common ancestor of a chimpanzee and a human virus. If you look, the same is true for HIV1 subgroup P, whose ancestor is C. The next ancestor along is a gorilla virus, and the same is true for HIV1 subgroup O. And there are a whole bunch of other chimpanzee viruses shown at the bottom, which didn't leave any descendants. So this shows that HIV1 there are at least 4 ancestors labeled A, B, C, and D, which were single viruses, which were the virus that was transferred from chimpanzees or gorillas to humans. And there's also a single common ancestor that's right at the root of this tree, which was a virus that was an ancestor to all of the different HIV-1 viruses and all of the different chimpanzee and simian viruses. Now, if you so those trees were all in the unit of time was in substitutions, but it's more natural to measure time in days or years. And to do that, you can take the phylogeny that we've been looking at, and you can say, if you have enough data to infer the rates of substitution, so if you know whether, here we've got 3 substitutions relative to the ancestor, and if you know 3 substitutions at the bottom, so both of the sampled viruses have 3 substitutions relative to the ancestor. And here, if we had a very strict molecular clock, which told us that we had one substitution every 2 days, you could date the events on this tree as being the split happening on day 2 and the overall ancestor being on day 0. And there are lots of software that obviously it helps if you have more samples, better data information, and so on. But there's lots of software that does this automatically, that encodes different sets of assumptions. The most popular by far is BEAST, and it comes in 2 flavors, BEAST 1 and BEAST 2. And there's tree data, tree time, and least squares dating that have the benefit of being computationally much faster than BEAST. They're probably less accurate in the process. So going back to the phylogenies, which I said we would go through at the beginning, this phylogeny is a dated phylogeny of a large representative sample of different HIV-1 viruses, HIV-1 subgroup M. And now we can read this phylogeny. So every tip on the phylogeny or leaf of the phylogeny has been rotated, so the present time is at the bottom and the root is at the top. So every viral sample that was put into this study is a tip of this tree, and you can notice some patterns. So, for example, a lot of samples seem to have come from a single study in 1983, because lots of tips are lined up with 1983, some are lined up with 1997, and the most recent samples were from 2001. One particular leaf has been highlighted, which was the Zaire 59 leaf, which is highlighted in black. So that's the oldest leaf in the study, and I guess. And as you go back, you can see lots of coalescences over time. So if we go back, for example, on the subtype B clade, you find a common ancestor, which is dated at roughly 1957. And if we go to the subtype C clade, you find a common ancestor that is roughly 1952. And if you go right back to the top of the tree, the common ancestor of everything in this tree is dated at roughly 1927 or thereabouts. So that would be the single virus from which all group M viruses descended. Now, one issue with inferring these phylogenies is that it's full of uncertainty. And how do we represent that uncertainty? Well, the simplest way to represent that uncertainty is by adding in numbers. So what we typically do in phylogenetics is you generate lots of trees, maybe under slightly different assumptions, or just statistical replicates using bootstraps, or if you're using something like BEAST, it'll be draws from a posterior distribution. Anyway, whichever way it is lots of trees. And then you choose one of your trees to plot, and you put numbers on this plot. And in this case, I put the number 60%. And what these 60% means, it means that in 60% of all of the trees that you've looked at, this common ancestor was found. There was an ancestor of only 2 of the viruses, these 2 viruses. These 2 viruses had a common ancestor, which is more recent than any of the other viruses in the sample. And it means that in 40% of the trees, actually, these 2 viruses were split by at least another virus, which had a more common ancestor within that picture. So if we look at the back at this tree here, you'll notice some numbers. So they've highlighted, the authors of this study have highlighted the nodes that had 100%, or where the support was 1. So these are nodes where 100% of the trees they looked at, this entire clade here, was separated from the rest of the tree in every single one of the trees. So they never found any ambiguity about this being the ancestor of these viruses and not being the ancestor of any of the other viruses in the study. So that's basically describing the phylogeny. Now just how do we start extracting information which is useful for epidemiology? Well, you start by noticing that we've focused on only the replication cycle, how a virus goes into a cell and then makes copies of itself. Well, interesting things may happen to viruses as they replicate. The virus may replicate, continue going from cell to cell, but at some point one of those virus particles or one of those cells may be transferred to another person, i.e. the virus is transmitted. So as you follow this lineage along the branch, at some point the virus continues to exist as of before, but it's inside a new person. Or if it is in a person, you may get migration. So in other words, the person who's hosting or carrying or infected with this virus may move to another location. So we can see examples of this in the real data. So in this diagram, you can see that I've highlighted that from E to F, there is a single branch at which the virus is likely to have jumped from person E to person F. And you can say that at this point, the viruses just kept on replicating cell to cell, but there was a point where that moved from one person to another. If we have a look at this tree, a real example of migration, you can say that at some point along the branch that I've highlighted, I think I've highlighted the wrong one, but at some point around these branches, the person or the people who are carrying this virus moved from Kinshasa to the Americas, because the root of the tree is in Kinshasa, and at some point the red color becomes the pink color. Obviously the resolution of this, these branches are very long, involving 25 years, so there was probably multiple transmissions and multiple migration events. But the end result is that at between there's a continuous lineage where viruses passed from cell to cell and person to person, migrated from Kinshasa towards the Americas. And there is a single founder for all of the viruses in the Americas that dates about 1955. All of the subsequent viruses.
Oh, I see.
They have too many differences, and so that's why branches are longer, which means that it takes longer time to achieve this, to accumulate these differences.
So far I've tried to make things hopefully as clear and simple as possible, and hopefully you found this useful. The one bit, as ever in a simple story, there's always complications. Some of the complications are sort of detail, statistics, and so on. There is one which is absolutely fundamental, which is recombination, because recombination doesn't just blur phylogeny, it actually messes up this tree-like structure, replacing the tree by an ancestral graph. So if you go back to our picture, you can imagine two viruses that separate and start to form lineages. There's one here at the top and one at the bottom. And if by chance a person gets co-infected with these viruses, and these viruses happen to infect a single cell, and I've given them both genotypes here, then you end up with a co-infected cell that carries two different viruses. So that's the first step, is co-infection. And then once you get a cell that's infected with viruses, you end up with double-stranded viruses that carry both of the genotypes. And then when they infect another cell, it actually takes 2 rounds of replication. The virus recombines, so the DNA you end up with in the next infected cell involves crossing over, and there's typically 10 crossing overs per genome when this happens. And here I've shown you the genome. The first part of the genome is the black one, is the AAC, and the bottom part of the genome is the GGAAC, and you end up with a virus which has got a discrete crossover linking together the 2 different genotypes. And we can sort of describe that and infer that, but the really unfortunate thing about this is that when it happens, we no longer have a tree when we're going backwards in time, because we have sort of coalescences and splits happening in both directions. Luckily, all is not hopeless. HIV is not the only biological replicator that recombines, and some other biological replicators, such as the one depicted here, have been very well studied in terms of their genotypes, and there are methods to either allow for recombination in phylogenies, so to obtain phylogenies that are approximately true, or in fact to infer the whole ancestral graph. So you can see as you go backwards in this tree, you have splits going in both directions. So in summary, phylogenetics provides powerful insights into the dynamics of virus spread at different scales. Trees are a natural way to describe ancestry, though recombination is still challenging. Ancestral state reconstruction is the key link to epidemiology. And when we've been, and I think a final important point I want to make, is when we've been describing sort of findings of PANGEA and so on these webinars, we've emphasized a lot the findings from our software phyloscanner, this software where we use these techniques to infer directly who infected whom. But there are many other tools and methods, including BEAST, the principal developer of which is Andrew Rambo, who's part of PANGEA and one of the PIs of PANGEA. And we should be using those tools, and we will be using those tools, and we should to analyze PANGEA and obtain many insights. And I think as we understand the phylogenies and do the ancestral state reconstruction and interpret that as epidemiology, we shall get a fuller picture looking at different scales. So here, to finish, thank you. What you see is the whole genome of one viral particle, so it's not a particularly long genome. The difficulty is that every genome from every particle in a host is different. This is in fact the first genome. This was a simple Nature paper, and there was the HXB2 genome generated in Paris, where the content of the paper said this is the genome of HIV, and printed these letters, and that was it. But I'll stop now and say thank you, and happy to take questions.